So here's something worth thinking about tonight. Right now, as you're lying there, settling in, getting comfortable, there are roughly 37 trillion cells in your body, give or take. And every single one of them is doing something. Right now, this very second. Little molecular machines humming along inside each cell, reading instructions from DNA, building proteins, shipping things from one compartment to another, burning fuel, dumping waste, sending signals. 37 trillion tiny living things cooperating so seamlessly that you don't even have to think about it. You just breathe. Your heart just beats. And all of that, every bit of it, traces back to one question that humans have been asking for as long as we've been capable of asking anything at all. How did life begin? Not how did humans evolve? Not how did fish crawl onto land? Not how did dinosaurs come and go? Those are incredible stories, and they have pretty good answers at this point. But the really deep question, the one that sits underneath all of those, is simpler and stranger. How did you go from a planet with nothing alive on it, just rock and water and gas and heat, to a planet with even one living thing? How do you get from chemistry to biology? From stuff that just sits there obeying the laws of physics to stuff that copies itself, feeds itself, and seems to want things? That's what we're going to talk about tonight. And I should tell you up front, nobody has the complete answer. Not yet. But what we do have, what's been pieced together over the last century or so, is genuinely breathtaking. There are pieces of this puzzle that fit together so beautifully, it'll make you smile. And there are pieces that are still missing, and the gaps are almost as fascinating as the answers. We're going to take our time with all of it. No rush. If you drift off somewhere along the way, that's perfectly fine. The story will still be here. Let's start at the very beginning. And I mean the very beginning. About 4.5 billion years ago, give or take a few tens of millions of years, Earth was not a place you'd want to visit. It had just finished forming, accreting, as the geologists say, which basically means it assembled itself out of a disk of dust and gas swirling around the young sun, chunks of rock and metal smashing into each other, sticking together, growing bigger, pulling in more material with their gravity. The whole process was violent, unimaginably violent. The surface of the early Earth was probably an ocean of magma, temperatures that would make a blast furnace look cozy. No atmosphere you'd recognize, certainly no oxygen, no ozone layer, nothing to block the ultraviolet radiation pouring in from the sun. And then, roughly 4.5 billion years ago, something truly catastrophic happened. A body roughly the size of Mars, scientists have named it Theia, slammed into the young Earth. Think about that. Not a meteor, not an asteroid. A planet-sized object hitting us at an angle at speeds of maybe 15,000 miles per hour. The collision was so energetic that it essentially liquefied both bodies. Huge amounts of debris were flung into orbit around what was left, and that debris eventually coalesced into the moon. Our moon. That quiet, familiar thing you see in the night sky? It's a scar from one of the most violent events in the history of the solar system. Now, why does this matter for the origin of life? Because it sets the stage. After the Theia impact, Earth had to basically start over. The surface was molten again. Any complex chemistry that might have been happening, gone. Reset to zero. The planet had to cool down, had to develop a solid crust, had to accumulate water on its surface before anything resembling life could even begin to be possible. And here's the thing. It did all of that surprisingly fast. Geologically speaking, anyway. Within a few 100 million years, Earth had cooled enough to have liquid water on its surface. We know this because of tiny crystals called zircons, little grains of zirconium silicate that are incredibly durable. Some of the oldest zircons ever found, from the Jack Hills region of Western Australia, are about 4.4 billion years old, and the chemistry locked inside those crystals tells us that liquid water was present on Earth's surface when they formed. 4.4 billion years ago. That's only about 150 million years after the planet itself came together. So you've got a young planet still getting hammered by leftover debris from the formation of the solar system. This period is sometimes called the Late Heavy Bombardment, though the exact timing and intensity of it are debated. But you've got liquid water. You've got an atmosphere, probably rich in carbon dioxide, nitrogen, maybe some methane, maybe some hydrogen. You've got energy pouring in from the sun, from volcanic activity, from lightning, from those very impacts. You've got all the raw ingredients: carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, the 6 elements that make up the overwhelming majority of all living things. They were all there. And somewhere in that chaotic, hellish, beautiful young world, chemistry started doing something unusual. Now, before we get into what that something was, let's talk about what we mean by life, because this turns out to be a surprisingly slippery question. You'd think it would be obvious. A dog is alive, a rock is not. But when you try to write down a precise definition, things get tricky fast. Fire consumes fuel, grows, and reproduces in a sense. Crystals grow and can even self-replicate their structure. Are they alive? Of course not. But why not? Most biologists, when pressed, will give you a list of properties rather than a single, clean definition. Living things metabolize. They take in energy and materials from their environment and use them to maintain themselves. Living things reproduce. They make copies of themselves. Living things evolve. Over generations, they change, they adapt, they diversify. And critically, living things carry information. They have some kind of instruction set, some molecular blueprint that gets passed from parent to offspring. That last one is really important for our story. Because when you think about the origin of life, you're really asking, how did the first information-carrying, self-replicating molecular system arise from a world that had no such thing? How did dumb chemistry become smart chemistry? And this is where a man named Charles Darwin enters, just briefly. Darwin, of course, is famous for the theory of evolution by natural selection, published in 1859 in On the Origin of Species. But Darwin was careful in that book. He barely touched the question of how life began. He knew it was a different problem. Evolution explains how life diversifies and adapts once it exists. It doesn't explain how it got started. But Darwin did think about it privately. In a letter to his friend Joseph Hooker in 1871, he wrote one of the most famous speculative passages in the history of science. He imagined, and I'm paraphrasing here, a warm little pond where all sorts of chemical compounds, ammonia, phosphoric salts, light, heat, electricity, might have come together to form a protein compound, which then underwent further changes. He was careful to call it speculation, but that image, the warm little pond, stuck. It became a kind of shorthand for the whole problem. For decades after Darwin, though, nobody really knew how to test the idea. The chemistry of life seemed so complex, so specific, so intricate, that it was hard to imagine how you'd even begin to study its origin in a laboratory. Proteins are made of amino acids, and there are 20 different amino acids used by life, and they have to be assembled in exactly the right order to fold into functional shapes. DNA is a double helix made of nucleotides, and the sequence of those nucleotides encodes all the information needed to build and run a living organism. How do you get from simple gases and minerals to that? It seemed like an impossible leap. And then, in 1952, a graduate student at the University of Chicago did something wonderfully audacious. His name was Stanley Miller, and he was 22 years old. His advisor was Miller-Urey, a Nobel laureate in chemistry who had been thinking about the composition of Earth's early atmosphere. Urey believed, based on the best understanding of the time, that the early atmosphere was what chemists call reducing, meaning it was rich in hydrogen-containing gases like methane, ammonia, and hydrogen itself, with water vapor but very little free oxygen. This was important because a reducing atmosphere is much more chemically reactive than the oxygen-rich atmosphere we have today. Oxygen, ironically, is actually quite destructive to complex organic molecules. It breaks things apart. A reducing atmosphere, on the other hand, is an environment where complex molecules can build up. So Miller, with Urey's guidance, built an apparatus. It was beautifully simple. A glass flask filled with water, representing the ocean, connected to another flask containing a mixture of gases, methane, ammonia, hydrogen, and water vapor, representing the atmosphere. He sealed the system, heated the water to produce steam, circulated the gases past a pair of electrodes that produced continuous electrical sparks, representing lightning, and then condensed the gases back into the water flask, creating a continuous cycle. Ocean, atmosphere, energy, circulation, a miniature early Earth in glass. He let it run for a week, and when he analyzed the water in the flask, it had turned a murky reddish brown, and dissolved in that murky water were amino acids, real amino acids, glycine, alanine, aspartic acid, several of the exact same amino acids that life uses to build proteins, from nothing but simple gases and a spark. Think about what that meant. In 1953, when Miller published his results, just weeks before Watson and Crick announced the structure of DNA, which is a remarkable coincidence of timing, he showed that the basic building blocks of life could form spontaneously from simple chemistry under conditions that might have existed on the early Earth. You didn't need a miracle. You didn't need some impossibly unlikely event. You just needed the right ingredients, the right energy, and a little time. The reaction was enormous. It was front-page news. The New York Times ran it. People were electrified. It felt like the origin of life was about to be solved. Maybe within a decade or two, scientists would figure out the whole pathway from simple molecules to living cells. That optimism, it turned out, was premature. Way premature. Because making amino acids is one thing. Getting from amino acids to a living cell is like saying, well, we figured out how to make bricks, so building a cathedral should be straightforward. The distance between those two things is almost incomprehensibly vast. But Miller's experiment did something that can't be overstated. It transformed the origin of life from a philosophical question into a scientific one. It said, "This is chemistry. This is something we can study, something we can test, something we can make progress on." And in the decades that followed, the picture got both more encouraging and more complicated. Other researchers repeated and extended Miller's work. They tried different gas mixtures, different energy sources, ultraviolet light instead of sparks, heat from simulated volcanic vents, even shock waves meant to mimic asteroid impacts. And they kept finding organic molecules, not just amino acids, but sugars, lipids, even some of the nucleotide bases that make up DNA and RNA. The building blocks of life, it seemed, were not hard to make. The universe was practically littered with them. We've since found amino acids in meteorites. The Murchison meteorite that fell in Australia in 1969 contained over 70 different amino acids. We've detected simple organic molecules in interstellar gas clouds, light years from any planet. The raw materials for life aren't rare or special. They're everywhere. But here's where the problem deepens and where the story gets really interesting. Because having a pile of bricks is not the same as having a building. Having amino acids floating around in a primordial soup is not the same as having a protein. And having a protein is not the same as having a cell. The question isn't really where the building blocks came from. That part we've got a pretty good handle on. The question is how those building blocks organized themselves into something that could sustain itself, copy itself, and begin the long, slow climb toward complexity. And to understand why that's such a hard problem, you need to appreciate something about the chemistry of life that isn't obvious until you really look at it closely. Living systems don't just use complex molecules. They use complex molecules that work together in interlocking cycles of mutual dependence. Proteins are built according to instructions encoded in DNA. But DNA can't do anything by itself. It needs proteins to read it, to copy it, to repair it. And both DNA and proteins need to be housed inside a membrane, a lipid bubble that keeps everything contained and creates a boundary between inside and outside, between the living system and its environment. So you've got this chicken and egg problem, or really, a chicken and egg and nest problem. Which came first? The proteins that do the work? The DNA that stores the information? The membrane that holds it all together? For a long time, this seemed like an impossible knot. And then, in the early 1980s, something happened that cracked the problem wide open, and it came from a direction that almost nobody expected. It came from the molecule that everyone had been overlooking, RNA. Now, you've probably heard of RNA. Most people know it vaguely as the middleman, the messenger, the thing that carries instructions from DNA out to the protein-building machinery. And that's true. RNA does do that. But for most of the 20th century, that's all anyone thought it did. RNA was the photocopy. DNA was the original blueprint. Proteins were the workers, and RNA was just the slip of paper that shuttled information between them. Important, sure, but not particularly interesting on its own. Nobody was looking at RNA and thinking, this is the key to everything. And then Tom Cech changed that. Thomas Cech was a biochemist at the University of Colorado, and in the early 1980s, he was studying a single-celled organism called tetrahymena. It's a little ciliated protozoan, the kind of thing you might find swimming around in pond water. Cech was interested in a particular process called RNA splicing, which is when a freshly made RNA molecule edits itself, cutting out certain sections and stitching the remaining pieces back together. This was known to happen, and everyone assumed that proteins, enzymes, were doing the cutting and stitching, because that's what enzymes do. They're the molecular machines that make chemistry happen in cells. So Cech was trying to isolate the protein enzyme responsible for this splicing reaction. And here's where it gets fascinating. He couldn't find it. He stripped away protein after protein, purified his system down to almost nothing, and the splicing kept happening. The RNA was cutting and rejoining itself all on its own, without any protein help whatsoever. At first, he thought he'd made a mistake. He thought there must be some trace amount of protein contamination that he wasn't detecting. So he repeated the experiment. He refined his techniques. He went to extraordinary lengths to eliminate every possible protein from the system. And still, the RNA spliced itself. What Cech had discovered was that RNA could act as an enzyme. It could catalyze a chemical reaction, in this case, its own rearrangement. This was genuinely shocking. Up until that moment, one of the bedrock assumptions of molecular biology was that only proteins could be enzymes. Only proteins had the structural complexity, the three-dimensional folding, the chemical versatility to speed up biochemical reactions. RNA was supposed to be a passive carrier of information, nothing more. And here was Cech showing that RNA could do chemistry, active, specific, catalytic chemistry. He published his findings in 1982, and almost simultaneously, Sidney Altman at Yale discovered something very similar. He found an RNA molecule in bacteria that could cut other RNA molecules, again functioning as a true catalyst, without any protein component. The two of them would share the Nobel Prize in Chemistry in 1989 for this work. And the molecules they discovered got a wonderful name: ribozymes. Part ribonucleic acid, part enzyme. Ribozymes. Now think about what that means for our chicken and egg problem. Remember the knot? DNA stores information but needs proteins to function. Proteins do the work but need DNA for their instructions. Neither makes sense without the other. But RNA can do both. It can store genetic information, just like DNA. Its sequence of bases encodes instructions, just like DNA does. And now, Cech and Altman had shown it could also catalyze chemical reactions, just like protein enzymes. One molecule, two jobs: information storage and chemical function, wrapped up in a single polymer. And that realization opened the door to one of the most elegant ideas in the entire history of biology: the RNA world. The term itself was coined by Walter Gilbert, a Nobel laureate at Harvard, in a short paper published in Nature in 1986. Gilbert's argument was beautifully simple. If RNA can both carry genetic information and perform catalysis, then maybe, just maybe, the earliest living systems didn't need DNA or proteins at all. Maybe the first self-replicating molecules on Earth were RNA. An RNA world, Gilbert called it. A stage in the history of life when RNA did everything, stored the genetic code, catalyzed the chemical reactions needed for survival, maybe even helped build the membranes that contained it all. DNA and proteins would have come later, gradually taking over the jobs that RNA did first, because they happened to do those specific jobs a little better. DNA is more chemically stable than RNA, so it became the preferred long-term information storage molecule. Proteins are more structurally versatile, so they became the preferred catalysts. But RNA was there first, doing both jobs adequately enough to get the whole thing started. And here's what's really lovely about this idea. It doesn't just solve the chicken and egg problem in the abstract. It actually has evidence behind it. Lots of it. Look at the molecular machinery inside your own cells right now. The ribosome, the structure that builds proteins by reading messenger RNA, is itself largely made of RNA. The catalytic core of the ribosome, the part that actually forms the chemical bonds linking amino acids together into a protein chain, that's RNA, not protein. RNA. The ribosome is a ribozyme, and this is true across all life on Earth, from bacteria to blue whales. The most fundamental piece of protein-building equipment in all of biology is, at its heart, an RNA machine. That looks an awful lot like a molecular fossil, a relic of a time when RNA ran the show. There are other clues too. The energy currency of your cells, ATP, adenosine triphosphate, the molecule that powers almost every energy-requiring process in your body, is built around a ribonucleotide. It's essentially an RNA building block with some extra phosphate groups tacked on. Many of the essential coenzymes that help protein enzymes do their jobs, molecules like NAD, FAD, coenzyme A, they all have RNA-like components at their core. It's as if the entire metabolic machinery of life was originally built from RNA parts, and then proteins were gradually layered on top. But the old RNA scaffolding was never fully removed. You can still see it if you know where to look. So the RNA World hypothesis is compelling. But, and this is important, it doesn't solve everything. It pushes the problem back a step, which is valuable, but it raises its own enormous questions. The biggest one is this: where did the first RNA molecules come from? Because RNA is not a simple molecule. Each nucleotide, each individual letter of the RNA code, is itself a fairly complex structure made of a sugar, a phosphate group, and a nitrogenous base, all joined together in a specific way. And then you need to string many of these nucleotides together into a long chain in a specific sequence before you have anything that could plausibly function as a ribozyme or a self-replicating molecule. Getting RNA to form spontaneously from simple chemicals on the early Earth, that's a genuinely hard problem. Some chemists have spent entire careers on it. For a while, this seemed like a potentially fatal objection. The individual components of RNA nucleotides, the sugars, the bases, the phosphates, could each be synthesized under plausible prebiotic conditions, but getting them to assemble into complete nucleotides was fiendishly difficult. The chemistry just didn't want to cooperate. You'd get the wrong isomers, the wrong linkages, tar-like mixtures of useless gunk. Through the 1990s and into the 2000s, this was sometimes called the prebiotic chemist's nightmare. And then along came John Sutherland. Sutherland is a British chemist, originally from the north of England, who spent years at the University of Manchester and later moved to the MRC Laboratory of Molecular Biology in Cambridge. the same institution, incidentally, where Watson and Crick worked out the structure of DNA. Sutherland approached the RNA nucleotide problem from a completely different angle than everyone before him. Instead of trying to make the sugar first, then the base, then stick them together, the way a chemist would logically build the molecule, he asked, what if prebiotic chemistry didn't follow that logical order? What if the components formed together, simultaneously, through a series of reactions where intermediates that don't look like any of the final parts gradually rearrange and converge into the finished nucleotide? In 2009, Sutherland and his team published a landmark paper in Nature showing exactly that. They demonstrated a plausible sequence of chemical reactions using simple starting materials, cyanamide, cyanoacetylene, glycolaldehyde, glyceraldehyde, and inorganic phosphate, that could produce 2 of the 4 RNA nucleotides through a pathway that never required assembling preformed sugar and base components. The phosphate, which everyone had assumed was just the backbone connector that came in at the end, actually played a crucial role throughout the synthesis, acting as a catalyst and a buffer at key steps. The chemistry was elegant. The conditions were plausible for the early Earth, and the yields were remarkably good. It was one of those results that makes you sit back and think, oh, maybe this really could have happened. Maybe the path from simple chemistry to RNA isn't as impossibly unlikely as we feared. Since that 2009 paper, Sutherland's group has extended the work dramatically. They've shown that similar networks of reactions, starting from hydrogen cyanide and a few other simple feedstock molecules, can produce not just RNA nucleotides, but also amino acids and lipid precursors, the building blocks of proteins and membranes, all from the same starting chemistry, under the same conditions. The implication is stunning. You don't need 3 separate miracles to get the 3 essential components of a cell. You might need just one connected network of chemistry, fed by a handful of simple molecules that would have been abundant on the early Earth. And this connects to something we touched on earlier: the energy sources available on young Earth. Sutherland's chemistry works particularly well with ultraviolet light as an energy input, which makes sense because the early Earth, lacking an ozone layer, was bathed in intense UV radiation from the sun. What was once seen as a destructive hazard, all that unshielded ultraviolet light, turns out to be potentially essential for driving the very chemistry that leads to life's building blocks. There's a beautiful symmetry in that, but knowing that RNA nucleotides can form is still a long way from having a self-replicating RNA molecule. You need those nucleotides to link up into long chains, and you need those chains to fold into shapes that can do something useful, specifically, copy themselves. And this is where the story takes us into some of the most creative, experimental work in modern science, because researchers have actually been trying to build self-replicating RNA systems in the laboratory, essentially trying to recreate in a test tube what might have happened on the early Earth billions of years ago. And the results have been both surprising and genuinely exciting. Let's talk about what they've found. So the challenge, right, is enormous. You need a strand of RNA that can act as an enzyme, a ribozyme, and specifically one that can copy RNA. Not just any RNA, but ideally itself. That's the holy grail. a molecule that reads its own sequence and produces a duplicate. And if you think about what that requires, it's staggering. The molecule has to grab free nucleotides floating around in solution. It has to recognize the complementary base on its own template strand. A pairs with U, G pairs with C. And it has to stitch those nucleotides together in the right order, one after another, hundreds of times, without making too many mistakes. That's a lot to ask of a molecule that has no brain, no proofreading machinery, none of the sophisticated protein enzymes that modern cells use. It's just a folded strand of RNA doing chemistry by virtue of its shape. The first big breakthrough came in the year 2001 from a lab led by David Bartle at MIT. Bartle's group had been working on this problem for years using a technique called in vitro evolution. Basically, they started with a huge pool of random RNA sequences, trillions of different molecules, and then they selected for the ones that showed any ability, however feeble, to join RNA nucleotides together. Then they took those winners, made copies with slight mutations, and selected again, round after round, generation after generation, mimicking natural selection in a test tube. And after many rounds of this, they produced a ribozyme they called the R18 polymerase. It was a piece of RNA about 189 nucleotides long, and it could actually extend an RNA template. It could read a short strand of RNA and string together complementary nucleotides, one by one, to build a new strand. Now here's the thing. R18 wasn't very good at it. It could add maybe 14 to 20 nucleotides before it stalled out or fell off the template. And remember, R18 itself was 189 nucleotides long, so it couldn't come close to copying something as long as itself. That's the fundamental problem. To be a true self-replicator, an RNA polymerase ribozyme needs to be able to copy a sequence at least as long as its own. Otherwise, it's like a photocopier that can only copy the first page of a 10-page document. Useful, maybe, but not self-sustaining. Still, R18 was proof of concept. It showed that RNA could, in principle, catalyze the template-directed synthesis of RNA. The chemistry was possible. The question was whether you could make it better. And people kept working on it. For over a decade, Bartle's lab and others refined and improved these RNA polymerase ribozymes. By 2011, Philip Holliger's group at the MRC Laboratory of Molecular Biology in Cambridge, the same institution, by the way, where Watson and Crick figured out the structure of DNA back in 1953, so there's a nice historical echo there. Holliger's group created a ribozyme called TC19Z that could copy RNA sequences up to about 95 nucleotides long. That's a huge improvement over 14. Not quite long enough to copy itself, but getting closer. And then, in 2016, Holliger's team made another leap. They engineered a ribozyme that could synthesize RNA strands longer than itself. Let me say that again, because it's kind of a milestone. They created an RNA molecule that could produce another RNA molecule longer than its own sequence. Now, it wasn't copying itself. It was copying a different, somewhat simpler template. But the length barrier had been broken. The photocopier could now handle a document bigger than its own instruction manual. But here's what's really interesting, and this is where the story gets more nuanced than just make a better copier. Some researchers started asking whether perfect self-replication was even the right goal to chase. Maybe the first replicators didn't need to copy themselves with high fidelity. Maybe the earliest RNA world was messier than that, more cooperative, more collective. Think about it this way. Imagine you don't have one super talented ribozyme that does everything. Instead, imagine you have a whole community of short RNA molecules, each doing something slightly different. Some are okay at copying, some are good at cutting, some help stabilize others. None of them is individually impressive, but together, as a network, they sustain each other. One molecule copies another, which copies a third, which copies the first. A kind of molecular ecosystem. This idea has a name. It's called a collectively autocatalytic set, and it was first proposed in a rigorous mathematical form by Stuart Kauffman back in the 1970s and 1980s. Kauffman, who's a theoretical biologist, argued that you don't need any single molecule to be a self-replicator. You just need a network of molecules where the set as a whole is self-sustaining. Each molecule's formation is catalyzed by some other molecule in the set, and there's experimental evidence that this can work with RNA. In 2009, the same year as Sutherland's nucleotide synthesis paper, actually, which is a nice coincidence, Tracy Lincoln and Gerald Joyce at the Scripps Research Institute demonstrated a pair of ribozymes that could replicate each other. Neither one copied itself. Instead, each one catalyzed the assembly of the other from shorter RNA fragments. Ribozyme A built ribozyme B, and ribozyme B built ribozyme A. And the system was self-sustaining. It could keep going indefinitely as long as you supplied the short RNA fragments as raw material. They even showed that the system could evolve. When they introduced variation into the fragments, some pairs replicated faster than others, and over time, the faster replicators took over the population. That's natural selection, happening in a test tube, with no proteins, no DNA, no cells. Just RNA. Now, you might be thinking, okay, but that system still needed pre-made RNA fragments to be fed into it. Where did those come from on the early Earth? And that's a fair point. But remember what we talked about with Sutherland's chemistry. Nucleotides can form from simple precursors, and short RNA strands can form spontaneously when nucleotides are concentrated on mineral surfaces or in evaporating pools. You don't need a factory. You just need the right conditions, and early Earth had billions of years and an entire planet's worth of real estate to try different conditions. And this brings us to one of the most important questions in the whole origin of life story. Where on earth did all of this happen? Where was the cradle? Because chemistry doesn't happen in the abstract. It happens in a place, in a physical environment, with specific temperatures and pressures and mineral surfaces and flows of energy and concentrations of reactants. And the environment matters enormously because it determines which reactions are possible and which aren't. For a long time, the default mental image was Darwin's warm little pond, a shallow pool on the surface, maybe near a volcano, warmed by geothermal heat, fed by rain-washing chemicals off the rocks, zapped by lightning, bathed in UV light. And there's a lot to recommend that picture, especially given Sutherland's UV-driven chemistry. But starting in the late 1980s, a completely different candidate emerged, and it changed the conversation dramatically. In 1977, scientists aboard the research submersible, Alvin discovered something astonishing on the floor of the Pacific Ocean, near the Galapagos Islands. They found hydrothermal vents, cracks in the ocean floor where superheated water, rich in dissolved minerals and gases, gushed up from deep within the Earth's crust. The water coming out of these vents was incredibly hot, sometimes over 350 degrees Celsius, and it was loaded with hydrogen sulfide, methane, hydrogen, and dissolved metals. And surrounding these vents, in the pitch darkness of the deep ocean, miles from any sunlight, there were thriving ecosystems, giant tube worms, clams, shrimp, microbial mats, all living not on solar energy, but on chemical energy from the vent fluids. The discovery was revolutionary for biology because it showed that life didn't need sunlight. It could run on chemistry alone. And pretty quickly, people started wondering whether life might have started at such vents. The idea was championed by several researchers, but one of the most influential was Günther Wechtershäuser, a German chemist and patent lawyer. Yes, patent lawyer, who in the late 1980s and early 1990s proposed what he called the iron-sulfur world hypothesis. Wechtershäuser argued that the first metabolic reactions happened on the surfaces of iron sulfide minerals, like pyrite, at hydrothermal vents. The idea was that these mineral surfaces provided both a catalyst and an energy source. Iron and sulfur could drive chemical reactions that built up organic molecules, step by step, right there on the mineral surface. No need for a pond, no need for UV light. Everything happens deep underwater, in the dark, powered by geochemistry. It was a bold idea, and it had some real chemistry behind it. Wechtershäuser showed that you could fix carbon dioxide, turn it into organic molecules, using iron sulfide minerals and hydrogen gas under hydrothermal conditions. That's significant because carbon fixation is one of the most fundamental metabolic reactions in all of biology. Every living thing needs to build organic molecules from simple carbon compounds, and here was a plausible way to do it without enzymes, without cells, without anything biological at all. Just rocks and hot water and dissolved gases. But the hydrothermal vent story got even more interesting in the early 2000s, when a different kind of vent was discovered. In the year 2000, an expedition led by Deborah Kelly from the University of Washington found a vent field in the mid-Atlantic that was unlike anything previously seen. They called it the Lost City, and the name was apt because it looked otherworldly. Towering white carbonate chimneys, some of them 60 meters tall, rising from the seafloor like the spires of some submerged cathedral. And the chemistry at Lost City was fundamentally different from the black smoker vents near the Galapagos. The water was cooler, around 40 to 90 degrees Celsius, hot, but not superheated. It was highly alkaline, with a pH around 9 to 11, and it was rich in hydrogen gas and methane, produced by a geological process called serpentinization, where seawater reacts with a type of rock called olivine in the ocean crust. And here's what caught the attention of origin of life researchers. Mike Russell, a geologist who had actually predicted the existence of alkaline vents years before Lost City was found, had been arguing since the late 1980s that this kind of environment was the ideal cradle for life. Russell's argument was elegant and specific. He pointed out that alkaline hydrothermal vents create natural proton gradients. Differences in hydrogen ion concentration between the alkaline vent fluid and the more acidic early ocean. And proton gradients, as it happens, are exactly what all living cells use to generate energy today. Every cell on Earth, from the bacteria in your gut to the neurons in your brain, makes its primary energy currency, ATP, by pushing protons across a membrane. It's called chemiosmosis, and it was discovered by Peter Mitchell, who won the Nobel Prize for it in 1978. And Russell's insight was that the very first cells didn't need to invent this mechanism from scratch. The vent gave it to them for free. The natural gradient between alkaline vent fluid and acidic ocean water could have driven the earliest energy-generating reactions long before cells evolved the sophisticated membrane proteins they use today. Think about what that means for a second. One of the most universal features of all life on Earth, the use of proton gradients for energy, might not be a biological invention at all. It might be a geological inheritance, a gift from the environment where life was born. The chemistry of the vent became the chemistry of the cell, and billions of years later, every living thing still carries that signature. Now, Russell and his collaborator, William Martin, took this further. They proposed that the porous mineral structures in alkaline vents, the tiny interconnected chambers in the carbonate chimneys, could have acted as natural compartments like primitive cell membranes. Each little pore was a tiny reaction chamber with thin mineral walls separating alkaline fluid from acidic ocean water, creating gradients that could drive chemistry. Organic molecules could accumulate inside these pores, concentrate, react with each other, and gradually become more complex. The vent in this picture isn't just providing energy and raw materials, it's providing structure, a physical scaffold for the earliest protocells. And this is where the story starts to get really interesting, because now you have two competing visions for the origin of life, each with passionate advocates, each with real evidence, and each with serious problems that haven't been fully solved. On one side, you've got the surface chemistry camp, people like John Sutherland, who showed that ultraviolet light from the young sun could drive the synthesis of nucleotides, amino acids, and lipid precursors all from the same starting materials. Chemistry happening in shallow ponds, warm little pools, maybe streams flowing across mineral-rich ground, everything bathed in UV radiation. On the other side, you've got the deep-sea alkaline vent camp, people like Mike Russell and Nick Lane, who argue that life needed a continuous flow of energy and that the proton gradients in places like Lost City provided exactly the right thermodynamic push to get metabolism going. And here's the thing. This isn't just an academic turf war. The disagreement cuts right to the heart of what we think life fundamentally is. Because depending on which camp you lean toward, you end up with a very different answer to the question: what came first, the information or the metabolism? The surface chemistry folks tend to emphasize replication. In their picture, the key event is the emergence of RNA molecules that can copy themselves, or at least catalyze useful reactions. And once you have that, natural selection kicks in and does the rest. The environment provides the raw materials. UV light provides the energy to cook them into interesting molecules. And then the magic happens when those molecules start behaving in ways that look like inheritance. Sutherland's work is beautiful because it shows you don't need separate origin stories for nucleotides and amino acids and lipids. They can all come from the same network of reactions, which makes the whole thing feel less like a series of impossible coincidences and more like chemistry doing what chemistry does when you give it the right conditions. But the vent folks push back on this, and their objection is worth sitting with for a moment. Nick Lane, who's a biochemist at University College London and one of the most eloquent advocates for the vent hypothesis, makes a point that I find genuinely compelling. He says, "Look, you can make all the organic molecules you want in a pond, but molecules sitting in a pond are at equilibrium. They're thermodynamically stable, and life is not an equilibrium system. Life is a far from equilibrium process. Every living cell is constantly burning energy to maintain its internal order, pushing against the natural tendency of things to fall apart and mix and reach equilibrium. The moment a cell stops doing that, the moment it stops pumping protons, stops maintaining gradients, stops actively keeping itself organized, it's dead. So the question isn't just, how do you make the building blocks? The question is, how do you sustain the energy flow that keeps those building blocks organized and reactive over time? And this is where the alkaline vents have a real advantage, at least in Lane's view. Because the vent doesn't just hand you molecules and walk away. It provides a continuous, sustained disequilibrium, a never-ending flow of chemical energy driven by the reaction between mantle rock and seawater. Serpentinization doesn't stop. It keeps going for tens of thousands of years, maybe hundreds of thousands of years, at a single vent system. That's a long time for chemistry to explore possibilities. And the proton gradient across those thin mineral walls, alkaline on one side, acidic on the other, that's not a one-time event. It's persistent. It's exactly the kind of sustained thermodynamic driving force that you need to push simple molecules uphill toward greater complexity. Now, you might be thinking, okay, but can't UV light also provide sustained energy? The sun was shining every day after all, and that's true. But Lane and Russell would argue there's a difference between energy that's available and energy that's coupled to chemistry in a useful way. Sunlight is incredibly energetic, but it's also pretty indiscriminate. It breaks bonds as easily as it makes them. UV radiation can build nucleotides, sure, Sutherland showed that, but it can also destroy them. There's a reason modern cells hide their DNA from UV light. It damages the very molecules that carry genetic information. So you've got this tension. The energy source that helps build your molecules is also the energy source that tears them apart. In a vent, the energy is gentler, more targeted. It's chemical energy, not radiant energy, and it's delivered in a way that's naturally coupled to the kinds of reactions that biology actually uses. But here's where it gets really interesting, because the debate doesn't stay neatly divided. Some of the most exciting recent work has started to blur the lines between these two camps, and I think that's where the real story is heading. Take the work of Dieter Braun and his group in Munich. Starting around 2007 and continuing through the 2010s, Braun showed something remarkable about what happens to molecules in thermal gradients. Situations where you have a warm side and a cool side in close proximity. If you have a thin crack in a rock, say, with warm water on one side and cooler water on the other, something fascinating happens. Convection currents form. The warm water rises, the cool water sinks, and dissolved molecules get carried along for the ride. But here's the clever part. Molecules also tend to drift toward the cold side through a process called thermophoresis. The combination of convection cycling molecules around and thermophoresis pushing them toward the cold side creates a trap. Molecules accumulate at the cold end of the crack, concentrating enormously, by factors of thousands or even millions compared to their original concentration in open water. Think about why that matters. One of the biggest problems with origin of life chemistry is concentration. In the open ocean, even if you're making interesting organic molecules, they're diluted in a vast volume of water. The chances of two RNA nucleotides bumping into each other and linking up are vanishingly small if they're floating around in an ocean. You need to concentrate them. Bronze thermal gradients do exactly that, and they do it passively. No biological machinery required, just physics. And here's what's wild. These thermal gradients exist in both surface environments and deep sea environments. You can find them in sun-heated rock pools near volcanic regions, and you can find them in the porous structures of hydrothermal vents. So this mechanism doesn't belong exclusively to either camp. It's a gift to both. Braun's group went further. They showed that thermal gradients could not only concentrate nucleotides, but could actually drive the formation of longer RNA strands. In experiments published around 2015, they demonstrated that cycling molecules through temperature gradients, repeatedly heating and cooling them, could promote the kind of strand separation and re-annealing that you need for something resembling replication. Warm temperatures melt double-stranded RNA apart. Cool temperatures let new strands form. It's a primitive version of what happens in a PCR machine, which biologists use every day to amplify DNA. But in Braun's experiments, no enzymes were needed. The thermal gradient did the work. So now you've got a physical mechanism that concentrates molecules, drives polymerization, and even promotes a crude form of replication, all without biology. And it works in exactly the kinds of geological settings that both camps are already talking about. That's a pretty big deal. But let's come back to the vent hypothesis for a moment, because there's another piece of evidence that Lane and others find particularly persuasive, and it has to do with a molecule you've already met in a different context. Remember when we talked about the core of the ribosome, how it's made almost entirely of RNA, and how that's one of the molecular fossils pointing back to an RNA world? Well, there's another molecular fossil that's equally fascinating, and it lives right at the heart of metabolism. It's called the acetyl-CoA pathway, and it's one of the oldest metabolic pathways in all of biology. The acetyl-CoA pathway is a way of fixing carbon, taking carbon dioxide from the environment and turning it into organic molecules. It's used today by certain archaea and bacteria, particularly those living in anaerobic environments. And what's remarkable about it is how simple it is compared to other carbon fixation pathways. It doesn't require light. It doesn't require oxygen. It uses hydrogen gas as an electron donor and carbon dioxide as a carbon source. And it runs the whole thing through a series of reactions involving iron-sulfur clusters and nickel, exactly the kinds of metal catalysts you'd find in the mineral walls of an alkaline vent. William Martin and others have argued that the acetyl-CoA pathway is the most ancient metabolic pathway, the one closest to the chemistry of the vent itself. In their view, the earliest protocells weren't photosynthetic. They weren't eating other organisms. They were doing something much simpler. They were using hydrogen and CO2 with mineral catalysts to build organic molecules from scratch. And the energy to drive this came from the proton gradient across the vent's mineral walls. Here's a detail that I love about this. If you look at the tree of life, the deepest branches, the ones closest to the last universal common ancestor, or LUCA, you find organisms that use exactly this kind of metabolism. They're methanogens and acetogens, archaea and bacteria that live on hydrogen and CO2 in anaerobic environments. They're not glamorous organisms. You won't find them in nature documentaries, but they might be the closest living relatives of whatever was alive 4 billion years ago. And they still use iron-sulfur clusters, they still use proton gradients, and they still run variations of the acetyl-CoA pathway. It's as if they never fully left the vent. Now, in 2016, Martin and a team of collaborators published a really striking paper where they tried to reconstruct the gene content of LUCA, the last universal common ancestor of all life. Not the first life, mind you, but the last organism from which all living things today descend. They combed through the genomes of thousands of modern organisms, looking for genes that were present in both bacteria and archaea, because if a gene is found in both of those deeply divergent domains, there's a good chance it was present in their common ancestor. And what they found was fascinating. LUCA's reconstructed genome pointed to an organism that was anaerobic, that used hydrogen as an energy source, that fixed carbon via the acetyl-CoA pathway, that depended on iron-sulfur clusters, and that used chemiosmosis, proton gradients across membranes, for energy conservation. In other words, LUCA looked exactly like what you'd expect if life had originated in an alkaline hydrothermal vent. But, and this is important, LUCA is not the origin of life. LUCA is already a fairly sophisticated cell with hundreds of genes, a genetic code, ribosomes, and complex metabolism. There's a vast stretch of evolution between whatever the first self-replicating chemistry was and LUCA. So even if LUCA lived in a vent-like environment, that doesn't necessarily mean life started there. Maybe life started in a surface pond, developed RNA replication, and then migrated to vents where it picked up chemiosmotic energy coupling. Maybe it started in a vent and later colonized surface environments. The evidence from LUCA is suggestive, but it's not conclusive, and honest researchers on both sides acknowledge that. And this is where a third possibility starts to creep into the conversation, one that's less about picking a single location and more about recognizing that early Earth was a dynamic, violent, interconnected place. Rain fell on volcanic landscapes and washed minerals into pools. Hydrothermal fluids percolated through cracks in the crust. Meteorites delivered organic molecules from space. UV light bathed everything above the waterline. And all of these environments were connected by water cycling through the crust, evaporating, condensing, flowing downhill, seeping underground. Maybe the origin of life wasn't a single event in a single place. Maybe it was a distributed process, with different steps happening in different environments, connected by the water cycle itself. Nucleotides forming in UV irradiated surface pools. concentration happening in thermal gradients within rock pores, energy coupling happening at vent interfaces, lipid membranes self-assembling wherever amphiphilic molecules accumulated at water-air boundaries. This is the kind of integrative picture that some researchers are starting to explore, and it has a certain elegance to it because it stops treating the origin of life as a single miracle and starts treating it as a network of interconnected chemical processes, each one plausible on its own, collectively capable of producing something extraordinary. And one of the people pushing this kind of thinking is someone we haven't talked about yet, a chemist whose work on protocells has opened up an entirely new way of thinking about what it means to be alive, Jack Szostak. And if you've been following along with the Nobel Prize thread running through this story, here's another one. Szostak shared the 2009 Nobel Prize in Physiology or Medicine, though that was for his earlier work on telomeres and the enzyme telomerase, the molecular machinery that protects the ends of your chromosomes. Brilliant work, hugely important for understanding aging and cancer. But that's not why he matters to us tonight. What matters to us is what Szostak turned his attention to afterward, or really, alongside, which is the question of how you get from chemistry to biology. How do you build a cell from scratch? And the reason Szostak's approach is so interesting is that he kind of sidesteps the old metabolism first versus replication first debate. He doesn't start with RNA. He doesn't start with energy gradients. He starts with the container, the membrane, the boundary that separates inside from outside. And his argument, which he's been developing since the early 2000s at Harvard and then Massachusetts General Hospital, is that you don't need sophisticated biology to get something that behaves remarkably like a living cell. You just need the right kind of membrane, the right kind of chemistry inside it, and the right kind of environment pushing the whole system forward. Let's talk about what he actually built, because it's genuinely beautiful work. Szostak and his collaborators, people like Katarzyna Adamala, Sheriff Mansi, and others over the years, focused on fatty acid vesicles, not the phospholipid bilayers that make up modern cell membranes. Those are sophisticated, tightly controlled structures that require enzymes to assemble. Fatty acid vesicles are much simpler. You take a fatty acid, oleic acid is the one they use a lot, dissolve it in water under the right pH conditions, and it spontaneously forms little spherical bubbles, vesicles. No enzymes needed, no biological machinery, just the physics of amphiphilic molecules doing what they naturally do in water. The hydrophobic tails cluster together, the hydrophilic heads face the water, and you get a bilayer membrane that closes into a sphere. It happens all by itself. Now, here's what makes fatty acid vesicles different from modern cell membranes, and why that difference is actually an advantage for thinking about early life. Modern phospholipid membranes are incredibly stable. They're tough. They're impermeable to most molecules, and that's great if you're a fully evolved cell with sophisticated transport proteins embedded in your membrane to shuttle things in and out. But if you're a primitive protocell with no proteins at all, an impermeable membrane is a prison. Nothing gets in, nothing gets out, you're dead in the water. Fatty acid membranes, though, they're permeable. Small molecules can slip through. Nucleotides, amino acids, simple sugars, They can diffuse across a fatty acid bilayer at rates that are actually meaningful for chemistry. So you get this wonderful situation where the membrane provides containment. It keeps your polymers inside, keeps your reaction products concentrated, but it doesn't cut you off from the outside world. You can still absorb raw materials from the environment without needing any transport machinery. Think about what that means. You've got a little bag of chemistry floating in a pond or sitting in a rock pour, and it can take in food and building blocks passively, just through diffusion. That's not nothing. That's actually a huge deal, because it means you can have a metabolism of sorts, chemical reactions happening inside the vesicle, without needing any of the elaborate molecular machinery that modern cells use to feed themselves. But Szostak went further. He showed that these fatty acid vesicles can grow. If you add more fatty acid molecules to the solution, they incorporate into existing vesicles, making them bigger. The membrane literally grows. And here's what's really clever. If you have two populations of vesicles, one containing RNA or some other polymer inside, and one empty, the ones with stuff inside them grow faster. They steal fatty acids from the empty vesicles. The reason is osmotic pressure. The RNA molecules trapped inside create osmotic stress on the membrane, which creates tension, and that tension is relieved by incorporating more fatty acid molecules. So the vesicles with more complex chemistry inside them outcompete the empty ones for membrane material. That's competition. That's a form of selection. And nobody designed it. It just falls out of the physics. And then there's division. If a vesicle grows large enough, it becomes mechanically unstable. It elongates into a long filamentous shape, and gentle shear forces, the kind you'd get from water flowing past or waves, or even just thermal convection, can break it into smaller daughter vesicles. Each daughter inherits some of the contents of the parent. So now you have growth and division, the two most fundamental properties of a living cell, happening without any biological machinery at all. Just fatty acids, water, and physics. Szostak published a particularly striking paper in 2009, the same year he won his Nobel, though for different work, showing this growth and division cycle in the lab. And in subsequent years, his group demonstrated that you could get RNA copying inside these vesicles. Not enzymatic copying, but non-enzymatic template-directed polymerization. You have a strand of RNA inside the vesicle. Nucleotides diffuse in through the permeable membrane, and they line up along the template and link together. It's slow. It's error-prone. But it works. And it works inside a membrane compartment, which means you've got the beginnings of heredity, information being copied inside a container that can grow and divide. Now, I want to be careful here, because this is not a living cell. Not even close. The copying is inefficient, the vesicles are fragile, the whole system requires carefully controlled laboratory conditions. Szostak himself is very clear about the gap between what he's built and anything you'd call alive. But what he's demonstrated is that you don't need to solve every problem at once. You don't need a fully functional RNA replicase before you can have selection. You don't need a perfect membrane before you can have compartmentalization. You can have crude, leaky, error-prone versions of all these things, and they can still work together in ways that look surprisingly lifelike. And this connects back to something we touched on earlier. the idea that life didn't spring into existence fully formed. It emerged gradually through a series of increasingly complex chemical systems, each one a little more lifelike than the last. Shostak's protocells give us a tangible picture of what one of those intermediate stages might have looked like. Not alive, exactly, but not dead either. Something in between. Something that grows, divides, competes for resources, and copies information, however crudely. Here's the thing that keeps me up at night about this, though. Well, not tonight. Tonight, you're supposed to be falling asleep. But the deep puzzle, even with Shostak's beautiful protocells, even with Sutherland's prebiotically plausible nucleotide synthesis, even with the thermal gradient work from Braun's lab concentrating and polymerizing molecules in rock pores, even with all of that, there's still this gap. This transition from chemistry that looks lifelike to chemistry that actually is life. And the gap is about information. Let me explain what I mean. In modern biology, DNA stores information. The sequence of bases, A, T, G, C, encodes the instructions for building proteins, and the proteins do the work of the cell. They catalyze reactions, build structures, transport molecules. The information in DNA is meaningful because it specifies functional proteins. But here's the problem for the origin of life. Random RNA sequences, the kind you'd get from non-enzymatic polymerization in a protocell, are mostly gibberish. They don't encode anything useful. They don't fold into functional ribozymes. They're just random strings of nucleotides. So how do you get from random sequences to sequences that actually do something? How does information enter the system? This is sometimes called the problem of functional sequence space, and it's genuinely hard. If you imagine all possible RNA sequences of a given length, say 100 nucleotides, the number of possibilities is 4 to the power of 100. That's a number so large, it makes the number of atoms in the observable universe look quaint. And the fraction of those sequences that fold into something functional, something that can catalyze a reaction or bind a specific molecule, is tiny, astronomically tiny. So how does random chemistry find those needles in that haystack? One answer comes from the work of people like Gerald Joyce, who we mentioned earlier, and also from a mathematician and theoretical biologist named Manfred Eigen. Eigen, working at the Max Planck Institute in Göttingen back in the 1970s, proposed something called the hypercycle, a theoretical framework for how self-replicating molecules could bootstrap themselves into existence. The basic idea is that you don't need one molecule that does everything. You need a network of molecules that help each other replicate. Molecule A helps replicate molecule B, which helps replicate molecule C, which helps replicate molecule A. A cycle. And Eigen showed mathematically that such cycles could emerge and stabilize even in a sea of random sequences, as long as the error rate of replication was below a certain threshold. That threshold is called the Eigen error catastrophe, or the error threshold, and it's a fascinating concept. Basically, if your copying fidelity is too low, if you make too many mistakes every time you replicate, then information can't be maintained. Each generation is too different from the last. Sequences drift randomly, and any functional sequence that arises by chance is immediately destroyed by copying errors in the next generation. It's like trying to preserve a message by playing telephone with 100 people who are all slightly drunk. the message dissolves into noise. So there's a minimum copying fidelity you need to maintain information, and that fidelity depends on the length of the sequence you're trying to preserve. Short sequences can tolerate more errors. Long sequences need more accurate copying. And this creates a catch-22, because long sequences are what you need for complex function. A ribozyme that's only 10 nucleotides long can't do much, but long sequences require accurate replication, and accurate replication requires sophisticated molecular machinery, which requires long sequences to encode. It's circular. Eigen himself recognized this problem, and it's one of the reasons the hypercycle model, while theoretically elegant, has been hard to connect to actual prebiotic chemistry. But here's where things get interesting again, and where the protocell work starts to matter in a new way. Because compartmentalization, putting molecules inside vesicles, changes the game. In a well-mixed open ocean, parasitic sequences would outcompete functional ones. A short, fast-replicating junk sequence would eat up all the nucleotides and drive functional ribozymes to extinction. But inside a vesicle, the population is small and contained. A vesicle that happens to contain a useful ribozyme, one that, say, helps copy RNA or catalyzes some beneficial reaction. That vesicle grows faster, divides more successfully, and its daughters inherit the useful ribozyme. Vesicles full of junk sequences don't grow as well and eventually lose out. So the vesicle acts as a unit of selection. It's not the individual RNA molecule competing anymore. It's the whole protocell. And this is exactly the kind of group selection that can maintain cooperation among molecules and prevent parasites from taking over. Urs Sathmary and John Maynard Smith, two towering figures in theoretical biology, worked this out in the 1990s in their stochastic corrector model. They showed that even without precise replication, even with significant errors, compartmentalized populations of replicators could maintain functional information and evolve. The math works. The compartment is the key. And Shostak's lab was building exactly those compartments. So now you can start to see how the pieces might fit together. Sutherland gives you the building blocks: nucleotides, amino acids, lipid precursors, all synthesized in a geochemically plausible surface environment. Braun gives you concentration and polymerization in thermal gradients. Shostak gives you the compartment, the fatty acid vesicle that grows and divides and allows crude replication inside. And the theoretical work of Eigen and Sathmary tells you that once you have compartmentalized replicators, natural selection can start to operate, even in a very primitive way, filtering for function, ratcheting up complexity one tiny step at a time. But we're still missing something. We've been talking almost entirely about RNA and membranes. We haven't really talked about the other great polymer of life, protein. And the relationship between RNA and protein is one of the deepest puzzles in all of biology, because in modern cells, they're utterly dependent on each other. RNA carries the instructions for making proteins, but proteins are needed to read those instructions. The ribosome, that ancient molecular machine we talked about earlier, the one whose core is made of RNA, is the place where these two worlds meet. And understanding how the ribosome came to be, how the genetic code itself was established, how translation evolved from something so primitive that it barely deserves the name, That might be the hardest question in origin of life research. Some scientists will tell you it's the hardest question. And tonight, lying here in the dark, let's see if we can at least trace the outlines of an answer. Start with what translation actually does, because it's easy to say RNA makes protein and leave it at that. But the process is staggeringly complex, even in the simplest bacterium alive today. You have a messenger RNA, a string of nucleotide letters, A, U, G, C, and the ribosome reads that string 3 letters at a time. Each triplet, each codon, specifies a particular amino acid. GCU means alanine. UUU means phenylalanine. AUG means methionine, and it also means start here. There are 64 possible 3-letter combinations and only 20 amino acids to assign them to. So some amino acids get multiple codons. There's redundancy built in, which turns out to be important. And the little adapter molecules that physically carry amino acids to the ribosome are transfer RNAs, tRNAs, small RNA molecules folded into a cloverleaf shape, each one with an anticodon on one end that matches a codon on the messenger RNA and an amino acid attached to the other end. The tRNA is the translator. It's the Rosetta Stone of the cell. It speaks both languages, the language of nucleic acids and the language of amino acids, and it bridges them. Now here's the thing that should make you pause. This system, as it exists today, requires dozens of protein enzymes just to function. The enzymes that attach the right amino acid to the right tRNA, the aminoacyl tRNA synthetases, are themselves proteins. So you need proteins to make proteins. That's the loop. That's the chicken and egg problem at its most acute. And the question is, how did this loop get started? Let's go back to the ribosome for a moment, because we touched on it earlier, but didn't really sit with it. The ribosome is enormous by molecular standards. It's made of 2 subunits, a large one and a small one, and together they contain about 50 different proteins and several long RNA molecules. In a modern bacterium like E. coli, the ribosome is roughly 2/3 RNA by mass and 1/3 protein. And here's the key discovery, the one that really changed the game. When scientists figured out the 3-dimensional structure of the ribosome at atomic resolution, and this was a monumental achievement. It took decades, and it earned Venkatraman Ramakrishnan, Thomas Stites, and Ada Yonath the Nobel Prize in Chemistry in 2009. When they finally saw the structure clearly, they found something remarkable. The catalytic heart of the ribosome, the peptidyl transferase center, where the actual chemical bond between amino acids is formed, is made entirely of RNA. No protein touches the active site. The proteins are on the outside, like scaffolding on a cathedral. They help the ribosome fold correctly. They stabilize its structure, but they don't do the chemistry. The RNA does the chemistry. The ribosome is a ribozyme. It's an RNA enzyme. And that's a fossil. That's a molecular fossil of the RNA world, sitting right at the center of the most important machine in all of biology. It tells you that before there were protein enzymes, RNA was catalyzing the formation of peptide bonds, the very bonds that link amino acids into proteins. RNA invented protein synthesis, and that means the first crude versions of translation didn't need protein enzymes at all. They could have been carried out by RNA alone. But how? How do you get from a world of RNA molecules doing simple chemistry to a system that reads a code and build specific proteins. This is where we have to be honest about how much remains uncertain, because nobody has a complete, step-by-step account of how translation evolved. But there are some genuinely beautiful ideas, and they've been gaining experimental support. One influential idea comes from thinking about what amino acids might have been doing around RNA before translation existed. And the answer is probably helping RNA. Michael Yaris at the University of Colorado spent decades studying the interactions between RNA and amino acids, and he found something really suggestive. When you look at RNA sequences called aptamers, short RNA molecules that have been selected in the lab to bind specific amino acids, you find a striking pattern. Aptamers that bind a particular amino acid tend to contain codons or anticodons associated with that amino acid in the modern genetic code. In other words, there seems to be a direct chemical affinity between certain RNA sequences and the amino acids they code for. The code isn't entirely arbitrary. There's a stereochemical foundation to at least part of it. Think about what that means. It means that in the RNA world, before any formal translation system existed, RNA molecules might have been interacting with amino acids in chemically specific ways. Amino acids might have been binding to RNA structures, maybe stabilizing certain folds, maybe acting as cofactors that helped ribozymes work better. And the particular amino acid that bound to a particular RNA sequence would have been determined, at least partly, by direct chemical recognition, by the shapes and charges of the molecules fitting together. Now imagine a very early, very crude version of a tRNA, not the elegant cloverleaf we see today, but maybe just a short RNA hairpin, a stem loop structure that happened to bind a specific amino acid at one end through this kind of stereochemical interaction. And imagine that this little RNA amino acid complex could interact with another RNA molecule, a primitive messenger, through base pairing at the other end. You'd have the bare bones of a translation system. No enzymes needed, no sophisticated machinery, just RNA molecules, recognizing each other through base pairing and presenting amino acids in a rough sequence. It would have been terrible. Let's be clear about that. The first translation would have been slow, error-prone, and the proteins it produced would have been short, sloppy peptides, maybe 5 or 10 amino acids long. But here's why even terrible translation might have been revolutionary. Even short, randomish peptides can do useful things. Some short peptides can stabilize membranes. Some can bind metal ions. Some can help RNA fold. And if a compartmentalized RNA system, one of Shostak's fatty acid vesicles, say, stumbled into producing even a slightly useful peptide, that vesicle would have had an advantage. It would grow a little faster, divide a little more reliably, survive a little longer, and natural selection would notice. This is the bootstrap. This is how you start climbing out of the RNA world and into the RNA protein world. You don't need accurate translation. You don't need a full genetic code. You just need a crude, noisy, barely functional system that occasionally produces a peptide that helps. And then selection refines it, one tiny improvement at a time. Ada Yonath, one of the ribosome Nobel laureates, has proposed something she calls the proto-ribosome. She looked at the modern ribosome structure and noticed a striking internal symmetry in the peptidyl transferase center. The region where peptide bonds are formed appears to be built from two similar RNA structures that are related to each other by a rotational symmetry, almost as if the original proto-ribosome was a dimer. Two copies of a simpler RNA molecule coming together. She and her colleagues have estimated that this ancestral structure might have been as small as a few dozen nucleotides, a tiny pocket of RNA that could catalyze peptide bond formation. That's all you need to start. And here's something else that's beautiful about this picture. Remember Eigen's error threshold from earlier? The idea that you can only maintain a genome of a certain length before copying errors destroy the information? Well, one of the things that protein enzymes do much better than ribozymes is catalyze reactions with high precision and speed. If the first crude peptides produced by prototranslation included even slightly better replicases, enzymes that copy RNA more accurately, then you'd have a positive feedback loop. Better translation produces better replicases, which copy RNA more faithfully, which allows longer genomes, which can encode better translation machinery. Each improvement enables the next. It's a ratchet. And once that ratchet starts clicking, you're on an escalator toward complexity. Lauren Williams and his group at Georgia Tech have been doing fascinating work on this, looking at the ribosome as a kind of geological record. They've developed what they call an accretion model of ribosome evolution. The idea that the ribosome grew outward from its ancient RNA core, adding layers of protein and RNA over time, like tree rings. The oldest parts are at the center, nearest the peptidyl transferase site, and they're almost entirely RNA. As you move outward, you encounter more and more protein. The newest additions, on the surface, are predominantly protein. It's as if you can read the history of the transition from the RNA world to the protein world by looking at the ribosome in cross-section. And the genetic code itself, how did it get refined? How did we end up with this particular assignment of codons to amino acids? Because the modern code isn't random, but it's also not entirely determined by stereochemistry. It has another remarkable property. It's error resistant. If you make a single letter mistake in a codon, if you misread one nucleotide, you're more likely to end up with a chemically similar amino acid than a wildly different one. Hydrophobic amino acids tend to be near other hydrophobic amino acids in the code. This means that translation errors, which must have been rampant in early life, would have been less catastrophic than they could have been. The code is optimized to minimize the damage from mistakes. How did that optimization happen? Probably through natural selection operating on populations of early cells with slightly different codon assignments. Cells whose codes were more error-tolerant produced more functional proteins and survived better. Over time, the code converged toward the near-universal version we see today. And it happened early, very early, because the code is essentially the same in every living thing on Earth, from bacteria in deep-sea vents to the cells in your own body. Whatever process established the genetic code, it was locked in before the last universal common ancestor, before the great divergence of life into bacteria, archaea, and eventually eukaryotes. And speaking of that divergence, let's think about what had to happen between the origin of the first crude protocell and LUCA, that last universal common ancestor we talked about earlier, the one Bill Martin's group reconstructed from comparative genomics. Because LUCA was already a sophisticated organism. It had DNA, not just RNA. It had a full genetic code. It had ribosomes. It had a complex metabolism built around the acetyl-CoA pathway. It used chemiosmotic coupling, proton gradients across membranes, to generate energy. LUCA was not simple, and the distance between a fatty acid vesicle with a few sloppy RNA molecules inside it and LUCA is enormous. It's like the distance between a campfire and a nuclear reactor. So what happened in between? One of the biggest transitions, maybe the biggest, was the switch from RNA genomes to DNA genomes. And this is interesting because DNA is chemically very similar to RNA. It's the same basic structure, just with one small modification. The sugar in the backbone is deoxyribose instead of ribose. It's missing one oxygen atom. That's it. But that tiny difference makes DNA dramatically more stable. RNA is fragile. It degrades easily. It's prone to hydrolysis. Its extra hydroxyl group makes it chemically reactive. DNA is tough. It can last for thousands of years under the right conditions. And if you're trying to store genetic information reliably, stability matters enormously. So the transition to DNA was probably driven by the advantage of having a more durable information storage molecule. RNA remained the working copy, the messenger, the catalyst, the regulator. But DNA became the archive, the master copy locked away and protected. And the enzymes that make DNA from RNA templates, reverse transcriptases, are ancient. They're found across all domains of life and in viruses. Some researchers have suggested that viruses might have actually invented DNA and then transferred the technology to cells, which is a wild idea, but not as far-fetched as it sounds. Patrick Forterre at the Pasteur Institute has been one of the strongest advocates of this hypothesis, pointing out that the enzyme cells used to make the building blocks of DNA, the deoxyribonucleotides, come in several chemically distinct varieties across different lineages, as if DNA synthesis was invented more than once. But whether DNA originated in cells or was borrowed from viruses, its adoption was a game changer. With DNA as the stable repository of genetic information and RNA as the functional intermediary, you get a division of labor that allows genomes to grow much larger. And larger genomes mean more genes, which means more proteins, which means more complex metabolism, and more sophisticated cellular machinery. The error threshold that Manfred Eigen worried about becomes less constraining when you have DNA polymerases that can copy with extraordinary fidelity. Modern DNA polymerases make roughly 1 error per billion nucleotides copied, which is almost unimaginably precise. And this brings us to another transition that had to happen somewhere along the way: the evolution of true cell membranes. Because Shostak's fatty acid vesicles are wonderful as protocells, but they have limitations. They're leaky, which is great when you want small molecules to diffuse in, but terrible when you want to maintain internal chemistry that's different from the outside environment. Modern cell membranes are made of phospholipids, which are more complex than simple fatty acids. They have 2 hydrophobic tails instead of 1, and a phosphate-containing head group, and they form much more stable, much less permeable bilayers. The transition from fatty acid membranes to phospholipid membranes must have happened gradually, and it must have co-evolved with the development of membrane transport proteins, specialized protein channels, and pumps that move specific molecules across the membrane in a controlled way. Because once your membrane becomes impermeable, you need doors, and those doors are proteins encoded by genes, built by ribosomes. Everything connects to everything else. And here's something genuinely puzzling that connects back to the deep-sea vent hypothesis we explored earlier. When you compare the membranes of bacteria and archaea, the two most ancient domains of life, they're chemically different. Bacterial membranes use fatty acids linked to glycerol through ester bonds, with one particular handedness of glycerol. Archaeal membranes use isoprenoid chains linked through ether bonds, with the opposite handedness of glycerol. These are fundamentally different membrane chemistries, and yet LUCA is the ancestor of both. So what kind of membrane did LUCA have? One possibility, championed by Bill Martin and others, is that LUCA didn't have a modern lipid membrane at all. Maybe LUCA was still using simple, mixed, leaky membranes. Maybe even mineral surfaces or inorganic compartments in hydrothermal vents served as its boundaries, and the two lineages independently evolved their own distinct membrane chemistries after they diverged. This is a controversial idea, but it fits with the picture of LUCA as something more primitive than any cell alive today. Not a free-living organism swimming around in the ocean, but a community of protocells nestled in the porous rock of an alkaline hydrothermal vent, dependent on the geological chemistry around it for energy and structure. If that's true, and it's a big if, but it's a fascinating if, then the emergence of free-living cells, cells that could leave the vent and survive on their own in the open ocean, was itself a major evolutionary achievement. It required inventing robust membranes, sophisticated transport systems, and self-sufficient metabolic pathways, and it might have happened twice, independently, once for the lineage that became bacteria, and once for the lineage that became archaea. Two separate escapes from the cradle. But let's pull back for a moment and think about timescales, because one of the most astonishing things about all of this is how fast it seems to have happened. The Earth formed about 4.5 billion years ago. The heaviest bombardment by asteroids and comets tapered off around 3.8 to 4 billion years ago, and the earliest evidence we have for life, carbon isotope signatures in ancient rocks from Greenland and northern Canada, possible microfossils in Australian chert, dates to somewhere between 3.5 and 3.8 billion years ago. Some researchers have pushed the evidence even earlier, to around 4.1 billion years ago, based on carbon isotope ratios in tiny zircon crystals from Western Australia, though that claim remains debated. But even taking the conservative estimate, it means that life appeared within a few 100 million years of conditions becoming even remotely hospitable. A few 100 million years sounds like a long time, and it is by human standards. But for the origin of something as complex as a self-replicating, metabolizing, membrane-bound cell, it's remarkably fast. It suggests that the origin of life might not be some wildly improbable fluke. It might be something that happens readily, given the right conditions and enough time. And that has implications that stretch far beyond Earth, implications that reach well beyond our own little planet. Because if life got started here that quickly, on a world that was still being pummeled by debris from the formation of the solar system, then what does that say about everywhere else? What does that say about all the other worlds, in all the other star systems, orbiting all the other suns in a galaxy that contains something like 200 billion of them? Let's sit with that question for a minute, because it's one of those places where the origin of life story stops being purely about chemistry and geology and starts brushing up against something almost philosophical. And yet, the way we approach it is still deeply rooted in everything we've been talking about: the chemistry, the energy sources, the membranes, the information systems. Because when astrobiologists think about where else life might have started, they're not just gazing at the stars and hoping. They're going back to those same questions we've been tracing all night. What does life need? What are the minimum requirements? And where else in the universe might those requirements be met? The short list is surprisingly specific and surprisingly common. You need liquid water, not ice, not steam, but liquid water, because it's the solvent that makes all this chemistry possible. You need an energy source, something to drive reactions away from equilibrium. You need the right elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, the CHNOPS, that make up virtually all biological molecules. And you need some kind of environment where concentrations can build up, where molecules aren't just endlessly diluted into nothingness. That's it. That's the starter kit. And here's the thing. Every single one of those ingredients is common in the universe. Water is everywhere. Carbon chemistry is everywhere. Energy sources are everywhere. The question isn't whether the ingredients exist elsewhere. It's whether they come together in the right way, in the right place, for long enough. And this is where our own solar system gets genuinely exciting, because we don't have to look to distant exoplanets to find promising candidates. We have them right next door. Take Mars, for instance. We know, not suspect, know, that Mars once had liquid water on its surface. Rivers carved channels into its terrain. Lakes filled craters. There may have been a shallow ocean covering much of the northern hemisphere. And this was happening around 4 billion years ago, right around the same time life was getting started on Earth. Mars had a thicker atmosphere then, warmer temperatures, active volcanism providing energy and cycling chemicals. It had everything on our short list. The Curiosity rover, poking around Gale Crater since 2012, has found organic molecules in Martian mudstones, not proof of life, but proof that the building blocks were there. It's found boron, which plays a role in stabilizing ribose, the sugar backbone of RNA. Remember Sutherland's work on prebiotic chemistry, how he showed that the same starting materials under UV light could produce nucleotides and amino acids and lipid precursors all at once. There's no obvious reason that same chemistry couldn't have played out on early Mars. The UV flux would have been there. The wet-dry cycling would have been there. The minerals would have been there. Now, Mars lost its magnetic field early on, maybe around 4 billion years ago. And without that magnetic shield, the solar wind gradually stripped away its atmosphere. The surface water evaporated or froze. The planet became the cold, dry, radiation-blasted desert we see today. But here's what's tantalizing. If life did get started on Mars during that early wet period, it might not have simply vanished. It might have retreated underground, into aquifers, into pore spaces in rock where liquid water might still exist today, kept warm by residual geothermal heat. We know that on Earth, there are thriving microbial communities kilometers below the surface, living in total darkness, feeding on chemical energy from rock-water reactions, completely independent of the sun. There's no reason in principle that something similar couldn't persist on Mars. The Perseverance rover, which landed in Jezero Crater in 2021, is specifically collecting rock samples from what was once a river delta, exactly the kind of environment where microbial life might have left traces. Those samples are meant to be returned to Earth eventually, where they can be analyzed with instruments far more sensitive than anything you can fit on a rover. It's one of the most ambitious sample return missions ever conceived, and it's fundamentally an origin of life experiment. We're asking, did it happen twice, independently, in the same solar system? But Mars isn't even the most exciting place to look. That distinction might belong to a moon. Specifically, Europa, one of Jupiter's 4 large Galilean moons, a world about the size of our own moon, covered in a shell of ice. But beneath that ice, a global ocean of liquid salt water, kept warm not by the sun, but by tidal heating. Jupiter's immense gravity flexes Europa as it orbits, squeezing and stretching the moon's interior, generating friction and heat. Enough heat to maintain a liquid water ocean, perhaps 100 kilometers deep. That's more water than all of Earth's oceans combined. And here's where it connects back to everything we talked about with hydrothermal vents. If Europa has a rocky seafloor, and there's good reason to think it does, then the interaction between that warm rock and the ocean water could produce hydrothermal vents. Alkaline vents, maybe, like Lost City. You'd have chemical gradients, mineral surfaces, dissolved hydrogen, carbon dioxide, all the ingredients that William Martin and Mike Russell argued could drive the emergence of metabolism. Europa might have its own version of the Lost City. Right now, at this very moment, churning away in the dark beneath kilometers of ice. The Europa Clipper mission, launched by NASA in 2024, is designed to investigate exactly this. It won't land, not yet, but it'll make dozens of close flybys, mapping the ice shell, measuring its thickness, looking for plumes of water vapor that might be erupting through cracks in the surface, and if it finds those plumes, analyzing what's in them. Because if water from that subsurface ocean is being sprayed into space, you could potentially fly through it and taste it. You could look for organic molecules, for chemical disequilibria, for signatures that something interesting is happening down below. It's an extraordinary prospect. And then there's Enceladus, a tiny moon of Saturn, only about 500 kilometers across, that turned out to be one of the most surprising objects in the solar system. The Cassini spacecraft, during its long mission orbiting Saturn from 2004 to 2017, discovered that Enceladus has geysers, jets of water ice and vapor erupting from cracks near its south pole, shooting hundreds of kilometers into space. Cassini flew through those plumes and found water, salt, silica nanoparticles, molecular hydrogen, and simple organic molecules. The silica nanoparticles are particularly telling because on Earth, they form in a very specific way, through hot water interacting with rock at temperatures above about 90 degrees Celsius. That's hydrothermal activity. Enceladus, this little ice ball that nobody was paying much attention to, has a subsurface ocean with hydrothermal vents. And the molecular hydrogen Cassini detected is exactly what you'd expect from serpentinization reactions, the same kind of rock-water chemistry that powers Lost City. The same chemistry that Martin and Russell proposed as the foundation of primordial metabolism. Think about what that means. On a moon of Saturn, right now, there is liquid water reacting with rock, producing the same chemical energy sources that may have powered the origin of life on Earth. We can't say life is there. We have no evidence of that yet. But the conditions look remarkably familiar. And this brings us to a concept that's been floating around astrobiology for decades, sometimes called the Copernican principle applied to biology. Copernicus showed us that Earth isn't the center of the solar system. Subsequent discoveries showed that our sun isn't the center of the galaxy, our galaxy isn't the center of the universe, and there's nothing particularly special about our cosmic address. The Copernican principle, loosely stated, is that we shouldn't assume we're special unless there's strong evidence that we are. Applied to life, it suggests that if the origin of life is a natural chemical process, not a miracle, not a 1 in a trillion accident, but a predictable outcome of certain conditions, then it should have happened elsewhere too. Maybe many elsewheres. Now, there's a famous counter-argument to this, and it's worth mentioning because it's genuinely unsettling. It's called the Fermi paradox, after the physicist Enrico Fermi, who supposedly asked over lunch one day in 1950, "Where is everybody?" If life arises readily, and our galaxy is billions of years old, and there are billions of potentially habitable worlds, then where are the signals? Where are the artifacts? Where's the evidence of anyone else? The silence is deafening, and people have proposed dozens of explanations, from the idea that intelligent life tends to destroy itself, to the possibility that we're simply not looking in the right way, to the more sobering suggestion that maybe the origin of life is in fact extraordinarily rare, and we really are alone, or nearly so. But here's what's interesting from the perspective of everything we've been discussing. The origin of life research of the last few decades has been steadily chipping away at the wildly improbable hypothesis. Every time someone like Sutherland shows that prebiotic chemistry is more plausible than we thought, every time someone like Sostak demonstrates that protocells can grow and divide without enzymes, every time someone finds that RNA can catalyze its own replication, or that thermal gradients can concentrate nucleotides, the needle moves a little further toward, this is just what happens when you have the right conditions. The chemistry wants to go there. It's not fighting against probability. It's rolling downhill. And that's a profound shift in how we think about our place in the universe. Because for most of human history, life was considered either divine or magical, or at the very least, utterly mysterious. Something that couldn't be explained by ordinary physical processes. And what the origin of life field has been doing, piece by painstaking piece, is showing that it can be explained. Not fully, not yet. There are still enormous gaps. We still don't know exactly how the first self-replicating system arose. We still don't have a complete scenario that goes from simple chemicals to a living cell without hand-waving at certain steps. But the trajectory is clear. The gaps are getting smaller, and the explanations are getting more detailed, more experimentally grounded, more chemically plausible. Which brings up one more thread worth following. The question of what we'd actually recognize as life if we found it somewhere else. Because everything we've talked about tonight has been life as we know it. Carbon-based, water-dependent, using DNA and RNA and proteins, running on the same genetic code. But is that the only way to do it? Could life be built on a completely different chemical foundation? Silicon instead of carbon? Ammonia instead of water? Some information-carrying polymer we've never imagined? This question, the question of alternative biochemistries, sits right at the edge of what origin of life research can tell us. And it's one of those places where the science starts to get genuinely speculative, because we don't have a second example of life to study. We've only ever found one kind. Every organism on Earth, from the bacteria living in boiling acid pools to the blue whale, shares the same fundamental chemistry. Same DNA, same RNA, same amino acids, same genetic code. We are all, without exception, one family. And that means when we try to imagine alternatives, we're doing it from a sample size of exactly one, which is a terrible position to be in if you're trying to figure out what's universal and what's just the way it happened to go here. But people have tried, and some of the ideas are genuinely fascinating, even if they remain deeply uncertain. Let's start with silicon, because that's the one everybody brings up. Carbon and silicon sit in the same column of the periodic table, which means they have the same number of outer electrons, 4, and can form the same number of bonds. Carbon is the backbone of every biological molecule we know. It links up with itself in long chains, rings, branching structures, and it bonds happily with hydrogen, oxygen, nitrogen, sulfur. It's spectacularly versatile. So the question is, could silicon do the same thing? And the short answer is, it's complicated. Silicon can form 4 bonds, sure, but silicon-silicon bonds are weaker than carbon-carbon bonds. They're less stable, especially in the presence of water and oxygen. Silicon dioxide, sand, basically, is a solid. Carbon dioxide is a gas. That difference matters enormously for chemistry. Carbon dioxide dissolves in water and participates in all kinds of reactions. Sand just sits there. Silicon also tends to form bonds with oxygen that are incredibly strong and hard to break, which means silicon chemistry in a water-oxygen environment tends to dead-end into silicates. Rocks, essentially. The molecules stop being reactive. They become geology instead of biology. Now, that doesn't completely rule silicon out. In a very different environment, maybe a world with no free oxygen, maybe at much higher temperatures where those silicon-silicon bonds become more manageable, maybe in a solvent other than water, silicon-based chemistry might have more room to play. Some astrobiologists have speculated about silicon biochemistry on worlds like Titan, Saturn's largest moon, where the surface temperature is around minus 180 degrees Celsius, and the lakes are liquid methane and ethane rather than water. At those temperatures, chemical reactions run incredibly slowly by our standards, but they do run. And silicon might behave differently in a hydrocarbon solvent than it does in water. Here's the thing, though. Even on Earth, biology does use silicon in limited ways. Diatoms, those gorgeous microscopic algae with intricate glass shells, build their structures out of silica. But the actual biochemistry, the information storage, the catalysis, the metabolism, that's all still carbon. Silicon is structural decoration, not the main act. So the honest assessment is that silicon-based life is conceivable in principle, but faces serious chemical hurdles that carbon doesn't, and we have zero evidence it's ever happened anywhere. What about the solvent? Water is so central to our biochemistry that it's easy to assume it's required. And water does have remarkable properties. It's liquid over a wide temperature range. It's an excellent solvent for ions and polar molecules. It has that weird density anomaly where ice floats, which insulates lakes and oceans from freezing solid. It participates directly in countless biochemical reactions: hydrolysis, condensation, photosynthesis. It's genuinely special stuff. But ammonia has some interesting properties too. Liquid ammonia can dissolve many of the same kinds of molecules water can. It has a wide liquid range under pressure. It's abundant in the outer solar system. The biochemist J.B.S. Haldane speculated about ammonia-based life back in the 1950s, and the idea has never fully gone away. The catch is that ammonia is liquid at much lower temperatures than water, around -33 to -78 Celsius at normal pressure, which means chemical reactions would proceed far more slowly. Life in liquid ammonia would be, by our standards, extraordinarily sluggish. But slow isn't the same as impossible. If you have billions of years, slow might be just fine. There are other candidate solvents too. Formamide, a simple organic liquid, has been suggested because it can dissolve nucleotides and even promote some of the same condensation reactions that are difficult in water. Supercritical carbon dioxide, sulfuric acid, which is relevant because Venus's clouds contain concentrated sulfuric acid droplets, and there's been a persistent, if controversial, thread of speculation about whether something might be alive up there. In 2020, a team led by Jane Greaves at Cardiff University announced the detection of phosphine gas in Venus's atmosphere, which on Earth is primarily produced by biological activity or industrial processes. The finding was intensely debated. Some groups couldn't reproduce it, others found methodological issues, and as of now, it remains unresolved. But it briefly electrified the astrobiology community precisely because it raised the question of life in an environment utterly unlike anything on Earth's surface. And then there's the deeper question, the one that really keeps origin of life researchers up at night. Forget the solvent. Forget the backbone element. What about the information system? Could life use something other than nucleic acids to store and transmit genetic information? This is where things get really interesting and really uncertain. We know that DNA and RNA work beautifully. The base pairing, the double helix, the ability to be copied and read and translated. But are they the only polymers that can do this? Almost certainly not. Chemists have synthesized what are called xenonucleic acids, XNAs, where the sugar backbone of DNA or RNA is replaced with something else: threose nucleic acid, TNA, hexatol nucleic acid, HNA, glycol nucleic acid, GNA. Several of these can form stable base pairs, can be copied by engineered polymerases, and can even be evolved in the lab to bind specific targets, just like natural nucleic acids. In 2012, Philip Holliger's group at the MRC Laboratory of Molecular Biology, the same Holliger whose RNA replication work we talked about earlier, demonstrated that 6 different XNA systems could store genetic information. be copied back and forth with DNA, and undergo Darwinian evolution. That was a landmark result because it showed that heredity and evolution are not unique to DNA and RNA. They're properties that can emerge from other chemistries too. Now, does that mean XNA-based life exists somewhere? We have no idea. But it means the chemical space for life is probably much larger than just the particular solution Earth landed on. And that's a humbling thought. It means that even if we understand perfectly how our kind of life began, we might be seeing only one tiny corner of what's possible. This connects to a concept that the physicist Lee Cronin at the University of Glasgow has been pushing: the idea of assembly theory. Cronin's argument is that we need a way to detect life that doesn't depend on recognizing specific molecules. Because if life somewhere else uses completely different chemistry, we wouldn't know what to look for. His approach is to measure the complexity of molecules, how many steps it takes to assemble them from simple building blocks. The idea is that very complex molecules that require many sequential steps to build, are unlikely to form without some kind of evolved, information-driven system. In other words, without life. So if you find a rock or an atmosphere or a liquid sample containing molecules above a certain complexity threshold, that's a biosignature, regardless of what those molecules are made of. It's chemistry agnostic. It doesn't care whether the life is carbon-based or silicon-based or something we've never imagined. It just asks, is this molecule too complex to have formed by chance? It's a provocative idea, and it's still being debated and refined. But it represents a shift in thinking that matters, a move away from looking for life as we know it, and toward looking for life as it could be. So where does all of this leave us? Let's take a breath and think about the full arc of what we've covered tonight. We started with a young Earth, barely cool enough to have liquid water, being pummeled by asteroids, wrapped in an atmosphere nothing like today's. And on that violent, hostile world, somehow, chemistry became biology. Simple molecules, hydrogen cyanide, formaldehyde, water, a few minerals, were coaxed by energy and circumstance into building blocks: amino acids, nucleotides, fatty acids. Stanley Miller showed us in 1953 that this first step wasn't as hard as everyone thought. John Sutherland showed us decades later that the pieces didn't have to be built separately and then assembled. They could emerge from shared chemistry, driven by ultraviolet light and wet-dry cycles, in a way that felt almost elegant. Those building blocks organized. RNA emerged as a molecule that could do double duty, storing information and catalyzing reactions. Tom Tech and Sidney Altman cracked that door open in the 1980s, and Walter Gilbert walked through it with the RNA World hypothesis. And then, over the following decades, researchers like David Bartle and Philip Holliger pushed the boundaries of what RNA could do, getting closer and closer to a self-replicating system, while Stuart Kaufman and others reminded us that maybe replication didn't have to start with a single molecule. Maybe it started with networks, with collective autocatalysis, with chemistry bootstrapping itself into complexity. We followed the debate about where it happened, warm little ponds versus deep sea vents, and saw how both sides had compelling arguments and real evidence. Bill Martin's reconstruction of LUCA as an anaerobic, hydrogen-dependent, vent-dwelling organism gave us a portrait of the last common ancestor that was startlingly specific and deeply strange. Jack Szostak showed us how simple fatty acid membranes could grow, divide, and house RNA without any proteins at all. Protocells that weren't alive exactly, but weren't not alive either. We watched the ribosome reveal its secrets through the work of Ramakrishnan, Stites, and Jonath, and saw that at its heart, this most essential machine of life is made of RNA, not protein, a fossil from the RNA world, still working, still translating the genetic code billions of years later. We traced the transition from RNA to DNA, the evolution of membranes, the divergence of bacteria and archaea, and the emergence of the genetic code itself, optimized against errors in a way that suggests it was shaped by natural selection before LUCA even existed. And then we looked outward. Mars, with its ancient rivers and perseverance patiently collecting samples. Europa, with its hidden ocean and the Clipper mission on its way. Enceladus, shooting plumes of salty, hydrogen-rich water into space, practically begging us to go take a closer look. Each of these worlds has the ingredients: liquid water, energy, the right elements, and if the origin of life really is chemistry rolling downhill, as the evidence increasingly suggests, then maybe it happened more than once. Maybe it's happening right now, somewhere we haven't looked yet. And that's the thought I want to leave you with as you drift off. Not an answer, because we don't have the answer yet, and honestly, we may not have it for a long time, but a trajectory, a direction. For the first time in human history, the question of how life began has moved from philosophy and theology into experimental science. Real people in real labs are doing real chemistry, and the results are converging on something that looks less and less like a miracle, and more like an inevitability. Not a simple inevitability. The details are staggeringly complex, and every answer raises 10 new questions. But the kind of inevitability that emerges when you have the right conditions, the right ingredients, and enough time. The chemistry wants to go there. It's been wanting to go there for 13.8 billion years, ever since the first hydrogen atoms formed after the Big Bang, and carbon was forged in the hearts of dying stars, and those atoms drifted through space and found each other on the surface of a small, warm, wet, rocky world orbiting an unremarkable star in an unremarkable galaxy. You are made of those atoms. Every carbon in your body was made inside a star. Every breath you take contains molecules that have been cycled through living things for billions of years. You are not separate from the story. You are the latest chapter of it. And somewhere out there, on a moon or a planet or a world we haven't discovered yet, the same story might be unfolding right now. Different characters, different chemistry, maybe a plot we wouldn't even recognize, but the same underlying theme: matter, given energy and time, organizing itself into something that can wonder about its own origins. That's enough for tonight. Let that thought settle. Let it be background noise, like rain on a window. The chemistry is still running. The experiments are still going. The spacecraft are still flying. And you can close your eyes knowing that the universe, in its quiet, patient way, is still figuring itself out, just like it always has been. Good night.